So yeah, it's a huge opportunity for me, both to be part of this center and also to tell you about it. So this is, you know, last year when I was here, we were just super excited about this possibility, and now for it to come through has just been absolutely amazing. So I'll just tell you very briefly, we've only got 15 minutes, but tell you briefly, you know, about many things that we're thinking about and doing, all under the context of trying to come up with a new way of understanding infection that involves computational modeling and measurement technologies. So I'm just showing you some blurbs from the grant that we wrote, application that we wrote. You can see here the idea really just to get into salmonella macrophage interactions in particular. I told you about the modeling, computation, and experiments. I also want to mention that one reason why I think this worked so well was that the Allen Foundation had identified modeling, measurement, and immunology as kind of being these key interest areas on the tour that Tom and Kathy went on to identify important interest areas. And we're really hoping that even though this is in salmonella and macrophages, that it has impact well beyond that. The fun thing about this is because of the Allen Foundation looking into this as opposed to the NIH, going back to these problems that you can have in academia, I was able to look a little more broadly for to find a great team. So we do have some PIs from Stanford and a domain expert in agent-based modeling from Virginia. But these two I want to talk about in more detail later. This is Mark Crady and Jerry Morrison. They're both from industry. And you know, one has a BS and one has an MS. Normally they wouldn't be thought of as PIs, but as you'll see in a minute, they're about as talented as could possibly be and fit very well into George's model of the kind of people you want in your life and on your team. Our whole project is based on a foundation of modeling, just a couple slides here on organization, so, and modeling on 3 different axes. So in my lab, we focus very much on trying to build models of cells, pathways, biological systems at that scale. Casey is an expert in biophysical modeling, so he looks at structures, and so it's a very, it's a different axis. And Shane is an expert in multicellular and agent-based modeling, and so that's the foundation. Now, what I think is really exciting is that we also have this computational layer, so that's what Mark and Jerry are expert in. And in particular, Mark and Jerry are people whose technology you guys use all the time. Okay, so Mark is an old friend. We actually had lockers next to each other at Jane Lathrop Stanford Middle School. Been friends ever since. So he, when I was going to grad school, he had this idea of trying to get the taxis and get basically position information from taxis and put that traffic information on a map that you could give to the world. So he made a company out of that that was quickly bought by Google, and then he and Jerry were the first to put maps for mobile on an iPhone, right? So all of us use that technology all the time. And they have this incredible expertise. And if you look at Jerry, I actually just found this out the other day. He was also on the original cut and paste with Electronic Arts. What makes these guys special is not only that they can deliver, right, but also that they think in terms of hundreds of millions of users, right, which I think, again, for academics is just, you know, many, many orders of magnitude different than what I think about. So I just love having them on the team. And again, very non-traditional, something that the NIH probably wouldn't have picked up. Because of their deep Google background, right, so we thought about what we wanted to do is very much like the Earth Engine, okay, which is the Google Earth Engine. Basically a way to do exactly what we want to do, take massive heterogeneous data sets, in this case from satellite imagery, et cetera, be able to wrap that in some kind of algorithms, right, computation and modeling, and then be able to let people use that to make new discoveries. And they've been incredibly successful doing this, so they have to figure out how to make the data available. They have to figure out the right tools that would be most applicable. But once they've done that, they have, you know, this API where people can get in and use it, and it's led to many interesting discoveries. So our idea was to somehow build an Allen cell engine, okay, where we're creating cutting-edge measurements. We're still wrapping it in these exciting algorithms, but now we're using them to direct biological discovery in some way. And again, at the largest scale possible. So the last thing I want to say is Shane, Casey, and I all do experiments, but we also have a domain expert in salmonella macrophage, and that's Denise Monak, and so it's been great to have her on board. So let me just take a second to talk about all three kind of legs, the measurement leg, the modeling leg, and the computation leg. So when it comes to measurement, what we're really interested in doing is trying to come up with quantitative dynamic, time-resolved, single-cell resolution type of experiments where you can really get the kind of data, the quality of data that you would need to model a system like this. And so we've been working in this space. Last year I talked about these kinase translocation reporters, which is just a simple synthetic construct that enables you to monitor the activity or the concentration of an active kinase in single living cells. I won't go into detail here. Happy to talk more in detail. But the fun thing is you can multiplex these. And so now what we can do, for example, here you can see on three axes, and I could have done more, but I don't have the slide space. But you can take something like a junk kinase and a P65, and you can look at them. This is now the same cell culture, right? And you can also look at salmonella at the same time. So if you look here, you can watch these infections, okay? And so you can go in and start to get a sense. And what we're particularly interested in is the fact that in some cells, you might have an infection with one salmonella bacteria, but nothing will happen. It will just sit there inertly for the entire course of the infection, whereas in a neighbor, it will grow out and overtake the entire cell and spread all of the bacteria further on in the culture. So how can we find any signals that correlate with this? Or more importantly, what seems to be what we're seeing is combinations of signals that matter. So as you can see right here. So here is our 3 different cells where we're looking at all 3 channels. And you can see some things right away. We're kind of still parsing through these data, but you can see that in the uninfected cells, they are responding in terms of an immune response. But one thing that's interesting is that P65 and the junk activity seem to be uncoupled. As you can see here, big peak, no peak there later. Whereas in the infected cells, you can see that there is a coupling, right? So it's one kind of simple observation that you can make. And we're really excited about trying to differentiate between these two, where the salmonella are growing out versus just staying inert that whole time. The other thing we've done is invented a new technology that wherein in the same cell you can get those kinds of dynamics that I just showed you, but you can also get the full RNA-seq transcriptome. And we've done this using Fluidigm systems where it's a microfluidics-based single-cell approach to getting gene expression. We've hooked it up to our microscope so that we're able now to get live cell imaging for individual cells, and we have to keep convincing our reviewers this because they often don't believe it. But this row is the dynamics of P65 in that cell, and this same row is the gene expression, which we could show you for 20,000 different transcripts. So we're getting heterogeneous data integrated into one cell simultaneously. Okay, quick about computation. Here what we're trying to do is just come up with the computational infrastructure and background, not only to be able to analyze all these kinds of data and kind of port it over into modeling, but also to make it accessible to everybody. I'll just show you a couple of things here. The biggest one, I'll give you a quote. It's a lab saying, but it actually comes from Michael, who's here today. When I was a young assistant professor, I sent over Michael an email saying, hey, I need some help with some image analysis. And I just got back this email that said, image analysis, oy vey. I don't know if you remember that. So that's a lab saying, image analysis, oy vey. It is a total disaster by and large, with every lab kind of trying to come up with their own best solution, which isn't really working. And I'm just showing here the flow of kind of what the state of the art is. And to mention that really throughout this whole process, there are hours and hours of human curation. So hundreds of hours really for a typical set of experiments that you might run. One of my postdocs recently asked himself if Facebook could recognize him, why couldn't he just automatically recognize these cells? And so in fact, that's what we do. We've just made a process that uses deep learning where we've converted image segmentation into image classification. I won't go into the details here, except to say that now we have something that can be used on bacteria, even in overgrown cultures. And you can see here with zero curation on our part, we're able to make those segmentations. And actually, so here I'm just showing you we can get these individual cell growth rates from that. But we can also do something that really I don't think anyone else in our space is doing. We can get automated cell cytoplasms from mammalian cells in essentially the same method. So it's an incredible new game changer for us. We're making the whole thing available. This is just saying you can even do multiple cell types in cold culture, which is exciting. But we're making the whole thing available to everybody. And the format that we've used, and Jerry's been working a lot on this, is just coming up with iPython notebooks. We were really excited when we saw the gravitational wave paper, not just because of the fundamental nature of the advance, but also because all of the data was available and ready to use in these iPython notebooks, right? So people had true access, and so that's really what we're working on a lot right now, is just giving people true, deep access into both our data and our methods. Okay, great. So in terms of the modeling, and again, this is just like a fire hose of information, right? But in terms of the modeling, so what our lab is best known for is building the first computer model of the simplest bacterial cell, Mycoplasma genitalium. And it was a cell that tried to take all of the known gene functions, all of the known molecular functions into account, right, to be predictive. That was super exciting for us, and it remains kind of a flagship technology for us. Very proud to see Cell recently announced that it was one of the highlights of their 40th anniversary. They put this up, and we were just thrilled about it. But now the question is, how can we build that technology from the simplest bacterium, which was already a big pain in the neck, get it now to something where we can do it for more complex or pathogenic bacteria and even their hosts at some point. So I'm proud to say that recently we've been working on an E. coli model. We don't have the whole cell, but we have something that accounts for something like 2,000 genes. It has an extensive new set of modeling things that we had to basically invent from scratch to make it happen. But there are many things now that we have, in particular with regard to regulation, on many levels. And very excitingly, it's able to integrate millions of different data points, all from different lab sources. It includes thousands and thousands of papers are part of this model and built in. Reminds me personally of Francis Crick's saying that they originally had this Project K, referring to the E. coli K12 strain, where they wanted to, and this was in the 70s already, where they wanted to have a complete solution of E. coli, right? Are we close to that? How can we go further? I think this model is really a first step to telling us what the direction should be. But there are many frontiers to kind of push through, and we are pushing on this space, especially because we're thinking about infection, right? Like host pathogen interactions, virulence, antibiotic response and resistance. I'll be really interested to hear the next talk. So there's many things to think about in this space. One thing that's been fun here is because we're also working on the measurement, we can try to make measurements that directly port in. So like for our kinase translocation reporters, they were made with a model in mind, and that model enables us to actually get quantitative characterization of the circuits that we examine. So we're able to not just measure and characterize, but really we describe those things mathematically, which has been exciting. Finally, just to talk about how we go from that to populations, I think that's one of the other key things, and it's been great to have Shane on the team. She's an agent-based expert, and she's also done more very complicated agent-based models. She is starting to build in technology so that we could push like that whole cell technology together with agent-based. And you could imagine making models of quorum sensing. You could imagine even trying to make models of bacteria and their hosts. And as you kind of put everything together that I've said, you know, now we can make these, generate these images of infection. We can extract all of the data. We can get individual growth rates from each bacteria that would be growing inside of some mammalian cell. We can then try to push that into both the whole cell setting and the agent-based setting to really try to get at some kind of large-scale quantitative description, and hopefully in that way come up with smarter ways to perturb and target that infection. So with that, I just want to say those are the three things we're considering, we're working on. It's been really exciting. I really want to thank the Allen Foundation, because it has just absolutely been amazing working with them, and the opportunities that they create for us, especially being able to bring on this computational team, has been really game-changing. So thanks. Okay. Yeah.
What do you see as the...
Markus, first, a great talk.
Oh, thanks.
What do you see as the gaps in the E. coli modeling that remain to be filled?
Yeah, so there's a whole bunch. Typically what we do is we just write out on the board all the functions that we see that are possible, and then we just start knocking them off. And in this case, what the model has is like the basic DNA replication, transcription, translation, but with all the regulation that would go in. Like our original model only ran for one division cycle. Right? It wouldn't be stable for many So now we have a model that is stable after 70, 80 generations. The next things that we really have to do that I see as big targets are spatiotemporal regulation, right? So that some of that stuff is known and it has to be included. I think a lot of the stuff related to interaction with other, for example, with bacteriophage or with other bacteria or with the host, I think those things are really important and have to be included. We'll target antibiotic mechanisms very soon here, and so you can imagine if we have all those mechanisms included, we can start, let's say, dialing down dosages and seeing what combinations of treatments, you know, very much inspired by some of the stuff you've done. So I think those are kind of key frontiers. And then, of course, there's all the stuff we don't know at all, right? But what we've even shown with this last model is that the model has a way of kind of telling you where to go next, you know. And I've been asked often by journalists, like, are you saying this is the end of experimentation? No, obviously. But I think a better goal and a very reachable one is no experiment wasted, right? So every single experiment actually has a reason, right? And it leads us to new knowledge that we can interpret. I think that's a fundamental goal of the lab. Yeah?
Large or significant fraction of genes in many bacteria are of unknown function. So
That's right.
I would think that having a very complete model of those that do have a function might give you some kind of angle on figuring out how those that have known function are acting. Is there a way to do that in a way that integrates with your models?
Yeah, that's exactly, we call that model-driven discovery. So the idea is to essentially, this is the way that I think about it, right? So, you know, since I've got brain people here, I'll throw out the old kind of, the old home tale, right? The average person only uses 10% of their brain cells or whatever. But I would say the average scientist uses several orders of magnitude less of their high throughput data, right? So, so what this model can do that's really exciting, right, is you can just, in a very fast way, you can generate a high throughput data set, generate the corresponding set of predictions, right? If the data and the model match up, great. We don't care about that so much because it means we already knew it. So you focus on the discrepancies because those tend to be highly enriched for new biology. And typically then what you can do is say, okay, well, what function, if it existed, would make these consistent with each other? And then that gives you a jumping point where you can start to explore.